Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Single-cell protein explained. Sources, production uses, and applications. Table of contents. Introduction to single-cell protein. History of single-cell protein. Organisms used as single-cell protein and the substrate used for their production. Fungi and substrates utilized by them and the protein content produced. Algae and substrates utilized by them and the protein content produced. Bacteria and substrates utilized by them and the protein content produced. Single-cell protein production method. Advantages of single-cell protein. Disadvantages of single-cell protein. Applications of single-cell protein. Introduction to single-cell protein. Single-cell protein refers to the dried cells of microorganisms that are consumed as a protein supplement by both humans and animals. The protein is obtained from microbial cells such as yeast, fungi, algae, and bacteria, which are cultivated on different carbon sources to enable protein synthesis. Due to the continuous increase in population and the global problem of nutrient deficiency, microbial biomass has gained importance as an alternative source of food and feed. SCP is rich in vitamins, including thiamine, riboflavin, pyridoxine, nicotinic acid, pantothenic acid, folic acid, biotin, cyanocobalamin, ascorbic acid, beta-carotene, and alpha-tocopherol. It provides essential amino acids, particularly lysine and methionine, which are vital for human and animal nutrition. In addition to proteins and amino acids, single-cell protein also contains minerals, nucleic acids, and lipids, contributing to its nutritional value. Microorganisms selected for single-cell protein production must possess certain important characteristics to ensure safe and efficient use. These include absence of pathogenicity and toxicity, making them safe for consumption, high protein content and good quality of protein, good digestibility and acceptable organoleptic properties, taste, texture, and smell, high growth rate to allow rapid biomass production, ability to adapt to unusual environmental conditions such as varying pH levels, temperature ranges, and mineral concentrations, capacity to utilize a wide range of carbon and nitrogen sources for growth and protein production. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. History of single-cell protein. The development of single-cell protein began during World War I. In 1919, Sack in Denmark and Hayduk in Germany introduced a method called Zulaufverfahren, where a sugar solution was continuously fed into an aerated suspension of yeast, instead of simply adding yeast to a diluted sugar solution. In Germany, Saccharomyces cerevisiae was produced from molasses to serve as a protein substitute. During the Second World War, microorganisms such as Candida arborea and Candida utilis were used for single-cell protein production. Specifically, Candida utilis was included in soups and sausages as a protein source during World War II. After the war, many industries were established in the USA and Europe, focusing particularly on the large-scale production of Candida utilis. During the 1960s and 1970s, single-cell protein production industries expanded across the UK, France, Italy, Russia, Japan, and Taiwan. The actual term, single-cell protein, was coined by Carol L. Wilson in 1966. In the 1960s, researchers at British Petroleum developed the proteins from oil process, a technology for SCP production. Early experimental work was carried out by Alfred Champagnat at BP's La Vera oil refinery in France, where a small pilot plant began operating in March 1963. For his pioneering contributions, Champagnat was awarded the UNESCO Science Prize in 1976. In the Soviet Union, large, single-cell protein plants producing Belkovo-vitamini concentrat, or protein vitamin concentrate, were set up alongside oil refineries, one in Kstovo, 1973, and another in Kirishi, 1974. By 1989, the Soviet Ministry of Microbiological Industry operated 8 plants, but growing environmentalist pressure led the government to shut them down or convert them to other microbiological processes. At the Second International Conference held at MIT in 1973, new findings were reported on the ability of actinomycetes and filamentous fungi to produce protein using various substrates. In more recent times, among the European communist countries, Russia maintained the highest single-cell protein production capacity, with at least 86 operational plants using different substrates. Organisms used as single-cell protein and the substrate used for their production. Microalgae. They are phototrophic organisms, considered potential single-cell protein due to their chemical composition, which includes proteins, essential fatty acids, especially omega-3, and various bioactive compounds. They have a relatively low nucleic acid content, 3 to 8%. Advantages: simple cultivation methods, effective utilization of solar energy, rapid growth, and high protein yield. Disadvantages: economical limitations in scaling up, digestibility problems due to the need for cell wall disruption, requirement of a large surface area for cultivation, and high contamination risk in open ponds. Examples: Tetraselmis suetica, Isochrysis galbana, Dunaliella, Tertiolecta, Chlorella stigmatophora, Spirulina species. Bacteria. Bacteria are considered potential single-cell protein because they possess high protein content, 50 to 80%, along with vitamins, phospholipids, and other functional molecules. They can grow on a wide range of substrates, including carbohydrates, gaseous, and liquid hydrocarbons. Advantages: ability to use many types of substrates, short generation time, and production of vitamins and micronutrients. Disadvantages: issues of palatability, high nucleic acid content, and possible production of toxins. Examples: Methylobacterium extorquens, Methylococcus capsulatus, Rhodobacter spheroides, Aphophila marina. Fungi, yeasts, and molds. Fungi can be divided into unicellular yeasts and molds. Yeasts, mainly used in aquaculture as they are protein-rich, with crude protein content of 38 to 52%. Molds, found to be highly digestible by fish. Advantages of yeasts: high malic acid content, ability to grow in acidic pH, and ease of harvesting. Advantages of molds: contain a relatively high nucleic acid content, up to 10%. Disadvantages: possible presence of toxins, slower growth rate, and lower protein content, 45 to 65%, compared to bacteria. Examples: yeasts, Saccharomyces, Cerevisiae, Cloiveromyces, Marxianus fungi, molds. Aspergillus oryzae, Yarrowia lipolytica. Different substrates used for fungi in single-cell protein production, along with their protein content. Maltose and glucose are used by Aspergillus fumigatus and Rhizopus chinensis. Cellulose and hemicellulose serve as substrates for Aspergillus niger, Aspergillus oryzae, Cephalosporium eichhorniae, and Chitomium cellulolyticum. Glucose, lactose, and galactose are utilized by Penicillium cyclopeum. Hydrocarbons are used by Yarrowia lipolytica and Candida tropicalis. Ethanol supports the growth of Candida utilis. Methanol is used by Pichia species and Pichia pastoris. Cellulose and pentose sugars serve as substrates for Cytilidium acidophilum and Trichoderma species, such as Trichoderma viridae and Trichoderma alba. Glucose is used by Fusarium venenatum with a protein yield of about 44%. Sulfite waste liquor supports Pecilomyces varioti and Candida utilis. Starch and glucose are used by Fusarium graminearum. Starch alone is utilized by Saccharomycopsis fibuligera, Candida utilis, and Saccharomyces cerevisiae. Banana waste, apple pomace, citrus pulp, potato starch processing waste, and waste liquor are used by Aspergillus niger, producing protein contents of 18, 17 to 20, 25.6, 38, and up to 50% respectively. Orange peel supports Aspergillus niger, Rhizopus oryzae, and Saccharomyces cerevisiae. Banana peel is used by Aspergillus terreus. Bergamot fruit peel is utilized by Penicillium roqueforti and Penicillium camemberti. Banana peel, pineapple peel, and papaya peel serve as substrates for Phanerochaetae chrysosporium and Panis tigrinus. Papaya waste, cucumber peelings, pomegranate rind, pineapple skin, and watermelon skin are used by Rhizopus oligosporus. Orange peel is also used by Trichoderma viride and Trichoderma ricei. Orange pulp and brewer's spent grain give a protein yield of 38.5% when processed with Saccharomyces cerevisiae. From dried potato and carrot skins, it yields 49.3%. From cucumber and orange peels, about 53.4%. And from discarded foods, nearly 39%. Cheese whey supports Candida krusei with a protein yield of 48%. Whey and potato pulp are used by Cluveromyces marxianus with about 33.7% protein. Juice, pulp, and peel from oranges and lemons, as well as cornstover effluent, are substrates for Rhodococcus opacus with yields between 42 and 52%. Molasses and bagasse are used by Candida tropicalis, producing 56% and 31% protein respectively. Rice bran serves as a substrate for many fungi, including Cladosporium cladosporioides, Penicillium citrinum, Aspergillus flavus, Aspergillus niger, Aspergillus ocraceus, Fusarium semitectum, and Monascus ruber, with protein contents ranging from 9 to 11%. Wheat straw is used by Pleurotus florida with a high protein content of 63%. Lignin serves as a substrate for Chrysonilla psittophylla with 39% protein. De-oiled rice bran is utilized by Aspergillus oryzae with 24% protein. Cheese whey filters are used by Trichoderma harzianum with 34% protein. Citrus pulp supports Trichoderma viridae with 32% protein. Waste capsicum powder, poultry litter, potato starch waste, and potato wastewater are used by Candida utilis, producing between 29 and 49% protein. Spoiled date palm fruit supports Hansennia spora uvarum and Zygosaccharomyces ruxi, both yielding 49% protein. Cheese whey and mixed agro-industrial wastes like orange pulp, molasses, brewer's grain, whey, potato pulp, and malt spent are substrates for Kaffir species, producing up to 54% protein. Breweries' spent grains serve as a substrate for Debariomyces hanseini, with 32% protein. Liquid sucrose is used by Hansenula jadini. Cheese whey and other agro-wastes also support Cloiveromyces marxianus, yielding 34 to 43% protein. Finally, inulin, crude, oil, glycerol waste, and hydrocarbons are used by Yaroia lipolytica, with yields ranging between 48 and 54% protein. Algae, the substrates they utilize, and their protein content. Anabaena cylindrica produces 43 to 56% protein. Aphanizomenon flosaquae contains about 62% protein. Arthrospira maxima yields 56 to 77% protein. Chlorella ellipsoidea has 42.2%, while Chlorella ovalis is lower, with 10.97% protein. Chlorella pyrenoidosa provides 57% protein, and Chlorella sperkii gives 6.87%. Chlorella vulgaris is rich, with 51 to 58% protein. Among the Dunaliella species, Dunaliella primilecta has 12.26%, Dunaliella salina gives 57%, and Dunaliella tertiolecta contains 11.4% protein. Porphyridium erugineum shows 31.6%, while Porphyridium cruentum ranges from 28 to 39% protein. Synodesmus almeriensis has 41.8%, and Synodesmus obliquus between 50 and 55% protein. Spirulina platensis produces 60 to 71% protein. Tetraselmis contains 36%, while Tetraselmis chewi varies between 31 and 46.5% protein. For special substrates, from soda ash effluent, Chaetomorpha antonina yields 14 to 18.2% protein. Ulva fasciata gives 13.7 to 18.6%, and Chlorella also grows here. When grown on tofu waste, tempeh waste, and cheese waste, different Chlorella species produce protein contents of 52.32%, 52%, and 15.43%, respectively. Chlorella salina from saline sewage effluents gives 51% protein. From natural habitats, Gracilaria domingensis has 6.2%, Gracilaria birdii 7.1%, Laurencia filiformis 18.3%, Laurencia intricate 4.6%, Chondrus crispus 20.1%, Porphyra umbilicalis ranges between 15 and 37%, and Gracilaria verrucosa ranges from 7 to 23% protein. From wastewater, Chlorella serociniana produces 45% protein, while Synodesmus obliquus reaches 52%. Spirulina cultivated in salinated and desalinated water yields 48.59% and 56.17%, respectively. Using CO2 and sunlight, Chlorella pyrenoidosa, Synodesmus quadricauda, and Spirulina maxima can produce up to 53% protein. Other examples include Nanochloropsis species, with 39.3%, Nanochloropsis gaudetana, also 39.3%, Desmodesmus species, 37.3%, and Schizochytrium species, ranging between 9.4 and 42.5% protein. Finally, Chlorella vulgaris can yield 17.9%, while Synodesmus species reach 48% protein. bacteria, the substrates they utilize, and the protein content they produce. Rhodococcus opacus grows on orange and lemon wastes. Aphophila marina strain STW181, when grown on commercial shrimp feed, yields more than 46% protein. From ramhorn substrate, Bacillus cereus produces 68% protein. Bacillus subtilis produces 71%, and Escherichia coli gives 66% protein. When cultivated on potato starch processing waste, Bacillus licheniformis produces 38% protein, and Bacillus pomilis yields 46%. From soybean hulls, Bacillus subtilis subspecies provides 26% protein. Using collagen meat-packing waste in fermenters, Bacillus megatherium is grown. Corynebacterium ammoniagenes grown on glucose and fructose yields 61% protein. Cellulomonas species and Alcaligenes species can utilize cellulose as a substrate. Lactobacillus bulgaricus and Candida cruzae grow well on whey. On synthetic growth medium, Cupriovitis necator produces 40 to 46% protein. Rhodops pseudomonas species yield about 45%. Rhodobacter capsulatus gives 23%. And Rhodops pseudomonas acidophylla produces 11% protein. From petrochemical wastewater, Haloarculus species IRU1 achieves 76% protein. Using methane from natural gas, Methylococcus capsulatus, Ralstonia species, and Brevibacillus agri yield 67 to 73% protein. Methylomonas and Methylophilus grown on sewage gas and liquid products produce less than 41% protein. On methanol substrate, Methylophilus methylotrophus and Methylomonas clara produce up to 81% protein. Methane also supports the growth of Methylomonas species, Methylococcus capsulatus, and some Trichoderma species. Acinetobacter chalcoaceticus uses ethanol, while Achromobacter delvacvatae can metabolize diesel oil in fermenters. Methylophilus species, grown on supernatant and biogas, give 24% protein. Methylomonas species grown on natural gas yield about 69.3% protein. From brewery wastewater, rhizospheric diazotrophs yield more than 55% protein. Rhodops pseudomonas belastica, using latex rubber wastewater, produces 66.7% protein. Rhodops pseudomonas palustris, cultivated in sludge or sago starch. Processing waste yields 72 to 74% protein. From poultry slaughterhouse wastewater, Rhodocyclus gelatinosus produces 67.6% protein. On pineapple waste, Rhodobacter spheroides strain P47 gives 66.6% protein. In fermented pineapple extract, Rhodops pseudomonas palustris strain P1 yields 65% protein. From soybean wastewater, Rhodobacter spheroides strain ZEET08 produces 52% protein. On glutamate malate medium, Rhodovolvum sulfatophyllum produces 15.6% protein. From pig farm waste, Rhodocyclus gelatinosus yields 50.6% protein. In sugar refinery wastewater, Rhodope pseudomonas and Rhodofulvum produce 58%. In miso-like effluent medium, Rhodocyclus gelatinosus gives 63% protein. From pineapple peel waste, Rhodobacter spheroides P47 produces again 66.6% protein. From seafood processing wastewater, Rhodocyclus gelatinosus yields 50% protein. On noodle wastewater, Rhodops pseudomonas palustris produces 50%. Rhodops pseudomonas species CSK01 from municipal wastewater yields 60.1% protein. From tuna condensate, Rhodocyclus gelatinosus strainer 7 produces 56%. From cassava waste, Rhodocyclus gelatinosus gives 45% protein. In simulated wastewater, Rhodopseudomonas palustris produces 45%, while from photosynthetic sludge, the same species reaches as high as 74% protein. Single-cell protein production method. 1. Selection of substrate and strain. Fast-growing microorganisms rich in protein are selected. Strains must possess suitable growth characteristics, such as safety, digestibility, and ability to use low-cost substrates. Suitable substrates, agro-wastes, industrial byproducts, or other low-cost carbon sources are chosen to support microbial growth. 2. Fermentation. The process is carried out in a fermenter designed for the mass culture of microbial cells. Microorganisms are inoculated into the medium and allowed to multiply. Key conditions such as temperature, pH, aeration, and oxygen supply are carefully controlled to maximize biomass yield and protein content. 3. Harvesting. After fermentation, microbial cells are separated from the growth medium. Techniques include centrifugation, filtration, or flocculation. A major challenge is the efficient harvesting and purification of biomass, which impacts cost effectiveness. 4. Post-harvest treatment. Harvested cells are dried to preserve them, reduce bulk, and improve storage stability. Drying methods may include spray drying, drum drying, or freeze drying. 5. Single-cell protein processing for food use. Dried microbial biomass undergoes further processing, removal of undesirable compounds, e.g., nucleic acids, toxins. Enhancement of nutritional quality, protein digestibility, vitamin enrichment. Improvement of sensory properties, flavor, texture, and color. Final products can be incorporated into animal feed or human food formulations. 6. Integration with circular economy. Optimal single-cell protein production emphasizes sustainable resource use. Low-cost agro-industrial wastes, methane, or wastewater can be used as substrates. This approach reduces environmental impact and supports a circular economy model by turning waste into high-value protein. Advantages of single-cell protein. Microorganisms grow much faster than protein-rich crops, which may take up to a year to produce. Single-cell protein provides high-quality protein with a content ranging between 60 to 80%. A wide variety of low-cost raw materials can be utilized as substrates. The overall production process is simple and efficient. Microorganisms can be genetically modified to enhance yield, nutritional value, or other traits. Production is not seasonal, allowing continuous availability throughout the year. Microbes can grow on a diverse range of substrates, including waste materials. Single-cell protein production is eco-friendly, cost-effective, and energy-efficient, making it a sustainable protein source. Disadvantages of single-cell protein. The production process is complex, requiring precise control of environmental factors such as temperature, pH, and aeration. Quality maintenance is difficult, as harvesting and purification after fermentation remain challenging. Single-cell protein does not provide a complete profile of essential amino acids, so it is usually consumed as a supplement rather than a primary protein source. Consumer acceptance is limited, as many people are hesitant to consume protein derived from microorganisms. Applications of single-cell protein. Animal feed and nutrition, used in poultry, pig, calf, and cattle feed to promote growth, fattening, and overall nutrition. Food industry, applied as food additives, vitamin carriers, aroma enhancers, and emulsifying agents to improve the nutritional value of baked goods, soups, and ready-to-eat meals. Single-cell protein is also used in starter cultures for bakers' yeast, brewing, and winemaking. Industrial uses, employed as a foam stabilizing agent and in processes like paper and leather manufacturing. Human nutrition and health, serves as an excellent protein supplement, especially for undernourished children, as it is rich in vitamins, amino acids, and minerals. Medical applications, incorporated in therapeutic and natural medicine to help manage obesity, diabetes, by lowering blood sugar, cholesterol, stress, and to prevent fat accumulation. Cosmetics, used in beauty and personal care products, such as herbal creams, lipsticks, and hair care formulations, due to its nourishing properties. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!